Hello children, in this video I will be dealing with the structure and function of enzymes. So with a small video clipping I will be giving commentary on the structure as well as function. So focus at the video and listen to the commentary. First let us define what is an enzyme. Enzymes are proteins that speed up chemical reactions in a cell. They are sensitive to temperature, pH and substrate concentration. Enzyme structure. There are basically two types of enzymes. Simple enzymes made up of proteins, and the second type is the conjugated enzymes or holoenzymes. They have two parts, the protein part as well as the non-protein part. The protein portion is called an apoenzyme and the non-protein portion is called a cofactor, and the cofactor can be of varying types. It can be a metallic cofactor like iron, copper, magnesium, or it can be an organic molecule like vitamins, and in that case it is known as coenzyme. It can also be a prosthetic group like peroxides. So there are three types of cofactors. Now here what is displayed is the three types of enzymes having the apoenzyme, the protein part, which is indicated in violet color. So you can see the violet part, the majority portion of the enzyme is the proteinous part, which is called the apoenzyme, and a small portion is the cofactor. It can be a metallic ion like iron, copper, magnesium, etc. as in the first case. In the second case you can see the violet color is the apoenzyme or the protein part, and here the cofactor is a vitamin like niacin. And the third case, it can be a combination, that is the apoenzyme with both the vitamin part as well as the metallic cofactor. Now in the textbook the third case is a prosthetic group, the prosthetic group like peroxides. So remember, apoenzyme is a proteinous part. It forms a major part of the conjugated enzyme, that is indicated in violet in all the cases, and only a small portion is the cofactor. The cofactor can be a metallic ion, or it can be a vitamin, or it can be a prosthetic group. Next, enzymes have an active site, a specialized region that is specific to certain substrates and binds with those substrates. And to explain this, there are two models. The binding of the substrate with the enzyme is described by two models. The first model is widely accepted and it is called the lock and key model. According to this model, the active site of the enzyme is static, stationary. The substrate is passive like a key, and the substrate like a key fits perfectly into the active site of the enzyme like the lock. So it's like a key fit into the lock. The second is the induced fit model. It is similar to the hand inside the gloves. The active site is flexible like the gloves. When we put our hand into the gloves becomes very flexible to fit in our hand. The substrate aligns the active site. It's like the hand getting into the gloves. But remember, once the reaction is over, the substrate, the enzyme reverts and gets its original shape and size, just like the gloves. When you remove your hand off the gloves gets its original shape. So here the enzyme active site is slightly flexible so as to make the substrate fit into the active site, whereas in the other case it is fixed. So remember, here the active site is flexible, not static, whereas in the previous model the active site is static. Now we can see the formation of the enzyme substrate complex. This is the enzyme with the active site. So just like the lock socket. Now the substrate like a key fits in to form the enzyme substrate complex, just like the lock and key. Now the specialty of this complex is that it lowers the activation energy required for the reaction to take place, and not only that, this complex or this interaction will weaken the bonds between the substrate, and as a result, see, the substrate is broken, the bonds become weak and it splits, and finally from the substrate two products are released, see, because the bond was weakened and now they are released. And then the enzyme reverts back to its original state. The enzyme is ultimately doing the same process over and over again. We need not form two products, it can form in one product. Now this is a very good example of the enzyme nitrogenase. This is with reference to chapter 12 of your textbook. It is figure 12.5. With reference to this figure, it is the action of the enzyme nitrogenase, and this particular enzyme catalyzes the conversion of the atmospheric nitrogen into two molecules of ammonia. Very, very popular example. We are learning right from the lower classes nitrogen fixation in leguminous plants, which is undertaken by the bacterium called Rhizobium. The enzyme responsible for this conversion of the atmospheric nitrogen into ammonia is the enzyme nitrogenase, which is indicated in yellow color. See how they have drawn the enzyme with the active site. Look at the shape of the active site. It is specific to the substrate, and here the substrate is nitrogen gas, which is drawn in such a way in proximity to the active site of nitrogenase. Now nitrogenase is two nitrogen atoms connected together with a triple bond. So now we are going to see first this nitrogen gas available in the atmosphere fits into the active site of nitrogenase enzyme. See, the second structure indicates the binding, that is to form the enzyme substrate complex. So here we see nitrogenase is bound with the substrate nitrogen, and remember nitrogen are two atoms connected with a triple bond. Now what happens is, first very important event that occurs is this complex lowers the activation energy, which is responsible for the reaction to take place. Not only that, it cleaves the bond between the atoms in the substrate. We know there are triple bonds or three bonds between the nitrogen atoms. You can see stepwise the bonds are diminishing or weakened. First, in the first step of reduction, the first bond between the nitrogen vanishes. Then the second step, the second bond vanishes, and the third step, the third bond or the triple bond vanishes between the two nitrogen atoms. And this is vanishing with the addition of hydrogen, and we know what is the process addition of hydrogen is called reduction. So we can first analyze each step. There are three steps of reduction. So we have the second structure indicating the complex enzyme substrate complex. Now the first step of reduction, two hydrogens are added. When two hydrogens are added of the triple bond, one bond between the nitrogen weakens or breaks, and what happens, the nitrogen binds to the hydrogen. So each nitrogen is bound to one hydrogen. Next, second step of reduction, again another two hydrogens are added. Now what happens, the second bond between the nitrogens vanishes or breaks, and each nitrogen is connected to two hydrogen atoms. Now in the third step, this two hydrogens are added. What happens? The bonds between the nitrogen, all three bonds disappears. There was initially triple bond, it became double bond, single bond, and now the entire all the bonds between the nitrogen vanishes, and each nitrogen is connected to three hydrogen. So nitrogen connected to three hydrogen, what is that molecule called? Ammonia. So now we can see two ammonia molecules are formed or attached to the nitrogenase. This was brought about by the catalyst or by the enzyme nitrogenase. It is acting as a catalyst. So we saw the three steps of reduction, and finally what happens, these two molecules of ammonia are released, and the nitrogenase is available to do the next cycle of events. it will catalyze the next nitrogen atoms and convert it into 2 molecules of ammonia. Again it repeats this procedure. So, I hope you have understood. First is indicating the nitrogenase enzyme with its active site. The second diagram indicates the binding of the substrate with the nitrogenase to form the enzyme substrate complex, and this is very relevant. It lowers the energy, and not only that, the bonds between the 2 nitrogen atoms of the substrate weakens, and that is stepwise. First step, 2 hydrogens are added, second step another 2 hydrogens are added, and third step the last 2 hydrogens are added. So, as each step proceeds, as we proceed from one step to the other step, what happens is the bond between the nitrogen decreases. From 3 it becomes 2, then 2 it becomes 1, and from 1 it becomes 0, and finally we have 2 molecules of ammonia which are liberated. This is how nitrogen of the atmosphere is fixed in the soil by these plants called leguminous plants, which has this particular enzyme called nitrogenase. So, with this you got a clarity on the structure and function of enzymes. Also connected to this, remember the fastest enzyme on earth is carbonic anhydrase, and we have already learned what is the role of carbonic anhydrase. In the RBC, it converts carbon dioxide in the presence of water to carbonic acid. So, this is the fastest enzyme on earth, carbonic anhydrase. Then second, the most abundant enzyme on the earth is rubisco, the enzyme which catalyzes both oxygenation and carboxylation, which is found in plants, both C3 and C4 plants. Only thing, their location is different. In C3 plants they are located in the mesophyll cells, whereas in C4 they are located in the bundle sheath cells. These are the most abundant, that is rubisco is the most abundant enzyme on earth. So, remember the 2 enzymes in addition to nitrogenase what we have studied now in this context. The fastest enzyme carbonic anhydrase and the most abundant enzyme rubisco. I hope you have understood this topic. Thank you.